I did my PhD at the University of Colorado, and I worked under Dr. Christy Anseth on therapeutic materials and developing polymeric hydrogels for therapeutic cell transplantation. Then I moved on to the University of Washington in Seattle, where I was able to explore a little bit more so using polymeric drug delivery systems, developing drug delivery systems for small interfering RNAs. And I worked with Dr. Pat Staten and Dr. Alan Hoffman at the University of Washington. And then I transitioned to my first faculty position at the University of Rochester. So I was hired as an assistant professor, and I'm now a professor of biomedical engineering at the University of Rochester, and also principal investigator at the Center for Musculoskeletal Research at the Medical Center. We are interested in developing therapeutic biomaterials, and for many different walks of life. Since this is an orthopedics audience, we have some very rigorous programs in developing targeted drug delivery systems to bone. And we're very interested in trying to target therapeutics to enhance the bone regenerative processes. In another area, we are interested in maybe the more traditional tissue engineering approaches where we can transplant cells and take advantage of three-dimensional hydrogel architectures to promote the recruitment of host tissue and the integration of those cells that we've transplanted to engineer bone. And so those are a couple of the areas that resonate for the orthopedics crowd. We also have active programs in developing tissue mimetics for salivary glands, actually, oddly enough, through some nice collaborations, as well as for the eye, which is a really interesting hierarchical tissue, and trying to develop this model so we can understand more about the disease of macular degeneration. And I should say that both of those projects are stemming from amazing collaborators that I have at the University of Rochester. Yeah, I mean, I think it's caused us to rethink a little bit in terms of, especially when labs were shut down. Certainly we were rethinking about, okay, in terms of our different research stories, should we be drafting up papers or writing reviews, or thinking about pivoting the projects in different realms. It's also caused us to really think carefully about experiments. So once labs reopened, as they are now, they're open, but in a reduced census in labs. And so we've really tried to think carefully about how do we design experiments that are most beneficial. I mean, not that we weren't doing that before, but I think it's really caused us to think very carefully about, what is the right next step, instead of what are the 6 next steps, because there's just limited time and resources to really address all of that at once. The other thing that I was certainly very interested in, but now, with the, interesting paradigm of the pandemic and limitations in the lab, interested in utilizing artificial intelligence and active learning to, build from what we know in the literature about, how materials interface with cells, how materials interface with the body, and use that to direct artificial intelligence type approaches and machine learning approaches that would instruct us what is the most effective next set of experiments that we could do that will tell us, a little bit more about the situation that we can then, continue to evolve, but do so in a less stepwise fashion. So really harness the collective knowledge that we have from the literature in a unbiased way, really, quite frankly, when using active learning approaches to direct that next best experiments, to understand more about the biophysical and biochemical cues that we can use, and how to develop the next greatest drug delivery system, or next greatest tissue engineering approach. So I think that that's something that is definitely spurred on, and ignited more so based on this idea of having to be more efficient in our approaches in the lab. Yeah, so in the two projects that I mentioned in terms of targeted drug delivery to bone, as well as tissue engineering approaches, what I hope to kind of drive towards in the next 5 years is certainly integrating and enlisting some of that learning tools to improve our efficiency, our experimental efficiency. On the other hand, I'm very interested in trying to delve closer into, clinical translation. So our targeted drug delivery system in particular, I think is, really unique, really powerful, and trying to take the next step in terms of, larger animal models, and then, maybe thinking about, starting a company regarding that technology and really trying to push it into the next realm. With regard to the tissue engineering approaches, the particular project that we have going on has to do with engineering the periosteum and understanding what the periosteum is doing during healing and recapitulating that. And one of my dreams is to be able to come up with an acellular approach. So we're operating under the auspices that the periosteum is there to support tissue ingrowth and recruit tissue ingrowth, but the cells that are embedded within the periosteum really don't play too large of a role in the long-term regeneration of that tissue, of that bone tissue. And so I would love to be able to, in 5 years, put a stamp on an acellular approach that would be as effective as some of the cellular approaches that we and others have developed. So those are kind of the next steps that I see, in the coming, half decade or so. So let's see how I do, how we do in the lab. I mean, I've got 2 young kids, so they keep me pretty busy, and they both love to ride their bikes around the neighborhood, which is something that I very much enjoy too. It's a little more leisurely these days with them in tow, but that's certainly really fun. I really enjoy baking. So my pandemic lifestyle has allowed me to make a regular muffin recipe every Sunday morning with my kids. And so we've been cycling through a variety of muffin types, and the family has decided that pumpkin chocolate chip is their favorite. So I certainly do a good amount of that. I have a garden. I love my garden. I'm looking forward to getting it going now that the snow has gone away. So that's also something that I really enjoy. I've been really lucky to have such a supportive society, and I have great friends and colleagues in the society, and they've been great collaborators. They've been great coaches and sponsors of me and my career. We have a really close relationship between the Center for Musculoskeletal Research at the University of Rochester and ORS. I participated when I was a young faculty. So again, in terms of professional development, when I was a young faculty, I participated in the ORS funded grant writing workshop. And I would definitely point to that as something that was key and critical in my development and success as a young investigator. And also the wonderful opportunities that ORS has given to my trainees. So I've had a couple of trainees that have won NERAs, so the New Investigator Research Awards. And so I see that as a really, very supportive in terms of my professional development when I was younger, when I was just starting out, as well as for the next generation. So my students that are coming up. through the ranks and supporting their professional development. So I think the ORS has remarkable programming at the annual meetings, but also all sorts of other types of meetings that are ongoing throughout the year, including the grant writing workshops that they put on. And those are just outstanding. And so I feel like with regard to professional development, my career would be really different in the absence of that support and amazing sponsorship that ORS provides its members. Yeah, you know, find something that is exciting to you, that you want to work on, and you know, put a lot of hard work in, have a lot of perseverance, and you'll get a lot out of it. So I think what's really unique about my lab is the opportunity to really delve into the synthesis of materials. So for, you know, drug delivery, tissue engineering, tissue models, what have you, and really getting, you know, the ability to make something, and then also, you know, not just make it willy-nilly, but have some design parameters that, you know, you hypothesize are going to enhance some elements of regeneration or mimicking tissue, depending upon what flavor, and testing it in vitro, testing it in vivo, and then coming back. So I think that that's a real value of my lab, is that, you know, we design for the application and we test, you know, through all sorts of different levels. And depending upon, you know, like, you know, all of my trainees get to touch all of those buckets. But typically, as is the case for and what I would suggest for a PhD in particular, is to go deep in one of those. And so, you know, whether you get really jazzed about being able to modify material properties and find out, you know, does the body care, which is often something we forget about, perhaps as engineers, but we try not to lose sight of that. Or maybe you're really interested in the in vivo and what's really happening in vivo when we do this modification, and so you can get really deep in one of those. So we offer breadth as well as depth, and really fantastic facilities and resources and collaborations. And the University of Rochester is a really great place to do orthopedic related research because of the easy access to the medical center, the outstanding environment, including the Center for Musculoskeletal Research. So it's a really great place to get to do our work.